I have been very interested in regenerative medicine since my graduate studies, where I read some papers about salamanders and how they actually are able to regenerate their arms. And then became actually pretty fascinated by the idea of regenerative medicine, because I think all of us are able to regenerate, and we show signs of that when we're first born. And when we're first born, we're actually able to have a lot of regenerative abilities, and we lose those over time. And those regenerative abilities are similar to those that I was originally reading about when I was in graduate school looking at salamander research. So I think that it's about trying to find what makes those different organisms be able to regenerate, and then harness our own ability to regenerate, and trying to be able to do that. So I was always fascinated by fibroblasts, because there is this group of fibroblasts at the base of our hair follicles, and each of our hair follicles have these fibroblasts at the base. And they're actually really interesting, because if you isolate these fibroblasts and you implant them underneath an area of skin that does not have hair follicles, such as the palm of your hands, those cells will actually induce the stem cells to create a hair follicle in the place that those fibroblasts are located. And I originally learned about those cells as a graduate student, and then I wanted to work on fibroblasts, so I came to Cambridge to do my postdoc, and I did my work in fibroblasts there, and subsequently took this position to pursue those interests. My laboratory's current research interest is in understanding why, when we're young, we have an ability to actually regenerate much better than we do when we're older. And that's seen in the skin as well as in our digit tips when we have wounds. And we repair and regenerate much better when we're younger. And right now we're trying to understand that particular process and be able to transfer that to an ability for us in later life. The clinical implications that the work that we're carrying out would be important for people with large wounds on their skin. Burns that cover a large percentage of the body are not able to regenerate their hair follicles and sebaceous glands and all the other structures that make up what we think of as skin. And it's important to be able to direct the stem cells to be able to reproduce those particular regions, which is currently not able to be done. So, so the implications for what we're trying to do is trying to understand how to direct those stem cells within our own body in order to recreate the structures that are lost in a wound. Okay, so the current challenges of the field are about, we're trying to be able to generate tissue on demand, in particular skin. It would be very nice to, in the future, to be able to have, to either grow skin in the lab on demand in order to treat patients that have large injuries. And secondly, also to be able to develop therapies for us to be able to regenerate our own wounds without having to rely on tissue within the lab. So right now, the most important thing is to be able to understand how to direct the stem cells that we already have in order to regenerate these particular regions. And even in, as far as we know, in adult skin, we have stem cells that can have the ability to regenerate. It's about directing them to regenerate in the right way. I think what would be really great in the next 5 to 10 years to be able to try to move the field forward is to be able to culture human skin inside the laboratory, and having all of the types of structures that are associated with skin that we have on our cells. And this would not only allow us to help people with large wounds that such as burn victims and in anyone with larger wounds, but also able to study human disease in vitro, which is something extremely difficult to do in single cell culture systems. So hopefully within the next, within the future, we'll be able to actually grow the tissue within the lab as it looks and is on our cells.